This is Lance Farside of GreenMedInfo.com bringing you another audio article. Amazing study finds body cells transfer genetic info directly into sperm cells. A revolutionary new study reveals that the core tenet of classical genetics is patently false, and by implication, what we do in this life, our diet, our mindset, our chemical exposures, can directly impact the DNA and health of future generations. A paradigm-shifting new study titled Somatic-to-germline transmission of RNA in mice xenografted with human tumor cells, possible transport by exosomes, promises to overturn several core tenets of classical genetics, including collapsing the timescale necessary for the transfer of genetic information through the germline of a species from hundreds of thousands of years to what amounts to real-time changes in biological systems. In classical genetics, Mendelian laws specify that the inheritance of traits passed from one generation to the next can only occur through sexual reproduction as information is passed down through the chromosomes of a species' germline cells via the egg and the sperm, and never through somatic or bodily cells. Genetic change, according to this deeply entrenched view, can take hundreds, thousands, even millions of generations to manifest. The new study, however, has uncovered a novel mechanism which somatic-to-germline transmission of genetic information is made possible. Mice grafted with human melanoma tumor cells genetically manipulated to express genes for a fluorescent tracer enzyme known as EGFP-encoded plasmid were found to release information-containing molecules containing the EGFP tracer into the animal's blood. Since EGFP is a non-human and non-murine expressed tracer, there was little doubt that the observed phenomenon was real. These EGFP trackable molecules included exosomes which were verified to deliver RNAs to mature sperm cells and remain stored there. The authors of the study pointed out that RNA of this kind has been found in mouse models to behave as a transgenerational determinant of inheritable epigenetic variations, and the spermazoal RNA can carry and deliver information that cause phenotypic variations in the progeny. The researchers concluded that their study's findings strongly suggest exosomes are the carriers of a flow of information from somatic cells to gametes, and that their results indicate that somatic RNA is transferred to sperm cells, which can therefore act as the final recipients of somatic cell-derived information. These findings overturn Weismann's genetic barrier, a principle proposed by the German evolutionary biologist August Weismann that states hereditary information can only move from genes to body cells and not the other way around. This idea has long been considered a nail in the coffin of the Lamarckian concept that an organism can pass on characteristics it's acquired during its lifetime to its offspring. Over the past decade, however, the seeming impenetrability of the Weismann barrier has increasingly been called into question due to a growing body of evidence that epigenetic patterns of gene expression such as histone modifications or gene silencing via methylation can be transferred across generations without requiring changes in the primary DNA sequences of our genomes, as well as the discovery that certain viruses contain the enzyme reverse transcriptase, which is capable of inscribing RNA-based information directly into our DNA, including germline cells, as is the case for endogenous retroviruses, which are believed responsible for about 5% of the nucleotide sequences in our genome. Nonetheless, as the authors of the new study point out, until their study, no instance of transmission of DNA or RNA-mediated information from somatic to germ cells has been reported as yet. The researchers further expanded on the implications of their findings. Work from our and other laboratories indicate that spermazoa act as vectors not only of their own genome, but also of foreign genetic information, based on their spontaneous ability to take up exogenous DNA and RNA molecules that are then delivered to oocytes at fertilization with the ensuing generation of phenotypically modified animals. In cases in which this has been thoroughly investigated, the sperm-delivered sequences have been seen to remain extrachromosomal and to be sexually transmitted to the next generation in a non-Mendelian fashion. The modes of genetic information delivered in this process are closely reminiscent of those operating in RNA-mediated paramutation inheritance, whereby RNA is the determinant of inheritable epigenetic variations. In conclusion, this work reveals that a flow of information can be transferred from the soma to the germline, escaping the principle of the Weismann barrier, which postulates that somatically acquired genetic variations cannot be transferred to the germline. The implications of research on exosome-mediated information transfer are wide-ranging. First, if your somatic cells, which are continually affected by your nutritional, environmental, lifestyle, and even mind-body processes, can transfer genetic information through exomes to the DNA within your germline cells, then your moment-to-moment decisions, behaviors, experiences, toxin and toxicant exposures can theoretically affect the biological destinies of your offspring and their offspring, stretching on into the distant future. Exome research also opens up promising possibilities in the realm of nutrigenomics and food as medicine. A recent study found common plant foods such as ginger, grapefruit, and grapes produce exomes that, following digestion, enter human blood undegraded and subsequently downregulate inflammatory pathways in the human body in a manner confirming some of their traditional folkloric medicinal uses. If the somatic cells within our body are capable, through extrachromosomal processes, of modulating fundamental genetic processes within the germline cells, or if foods that we eat are also capable of acting as vectors of gene regulatory information, truly the old reductionist, mechanistic, unilinear models of genetics must be abandoned in favor of a view that accounts for the vital importance of all our decisions, nutritional factors, environmental exposures, etc., in determining the course not only of our bodily health, but the health of countless future generations as well. Thanks for listening. This is Lance Farsight for GreenMedInfo.com, bringing you another informative article. Make sure to visit our website and subscribe to this channel for more information to stay updated on your healthy lifestyle.